An individual's phenotype matches their genotype, the genetic makeup, right? Well, maybe not all the time. Hi, biology friends. It's Sarah, your other biology professor, and let's talk about penetrance and expressivity. Let's imagine we've got a population of plants that produces flowers, and in this population, the big R, red flower allele, is dominant over the little r, white flower allele. And we sample all the heterozygotes in this population, and we find that every single one produces red flowers. So 100% of individuals with the big R, little r, red genotype produce the expected red phenotype. This is complete or full penetrance. Penetrance is how frequently you see the expected phenotype for individuals with a particular genotype. You flip a switch, you expect to see light. You've got a big R allele, you expect to see red flowers. Imagine instead that 20 individuals with the big R, little r genotype are tested, and we find that 17 of them produce red flowers and the others produce white flowers. 17 out of 20 produce the expected phenotype based on their genotype. That's a penetrance of 85%. So we could call this incomplete or reduced penetrance. Incomplete penetrance occurs when some individuals with a particular genotype express the expected phenotype, but others don't. The genotype isn't always penetrating through to be expressed as the phenotype. Why? Well, there are some reasons for that, and we'll get to those later. But for now, let's look at a human example. Huntington's disease is an autosomal dominant neurodegenerative disorder caused by repeat expansion. The sequence CAG in the gene encoding the Huntington protein has gotten way longer. All individuals with 40 or more CAG repeats will develop Huntington's disease. So for individuals with genotypes containing a 40 repeat allele, Huntington's disease is completely penetrant. But individuals with between 36 and 39 CAG repeats may develop Huntington's disease, or they may not. Incomplete penetrance. One study showed a penetrance of 51% for genotypes containing the 38 repeat CAG allele, meaning that individuals with that 38 repeat allele have a 51% chance of developing Huntington's disease by age 75. So the penetrance can also tell you the probability that an individual with a particular genotype will develop or express a particular disease or phenotype. Maybe an individual with a certain genotype does express the expected phenotype, but a more or less exaggerated version of that phenotype. That gets us to expressivity, the degree to which individuals with the same genotype express the phenotype. If we go back to our red flowers and the 17 individuals that expressed red flowers, to what extent? Expressivity could be constant, meaning all 17 individuals express the phenotype in the same way, the same intensity of red color. Or some individuals could have expressed the red as a lighter red, while others a bold, deep red. That's variable expressivity. With variable expressivity, the phenotype is expressed. The flowers aren't white, they're red. But the degree to which the phenotype is expressed, how intense that red is, the range of reds, is varying between individuals. So here, the light's on, but how intense is it? For a disease phenotype that shows variable expressivity, individuals with the same genotype could show mild, moderate, or severe forms of that disease. Not a disease, but a clinical example, polydactyly in humans, extra fingers and toes, is autosomal dominant and shows both incomplete penetrance as well as variable expressivity. One form, where the extra digit is on the same side as the little finger, shows a penetrance of around 65%, depending on the population tested, and variable expressivity in terms of the number of extra digits and how well developed they are. Why might the genotype not perfectly match the associated phenotype 100% of the time? Well, if you've studied the processes of gene expression, like transcription and translation, you know that there are many steps involved. And that's a lot of steps at which something else can come in and interfere. So at any point between gene and functional product, something like environmental factors, mutations in gene regulation, or the products of other genes can have an impact on that finished product. To sum up these often mixed up terms, when you think penetrance, think the other P words, percentage and probability. Penetrance is measurable. What percentage of individuals with a particular genotype show the expected phenotype? 100% complete penetrance. Less, incomplete penetrance. And penetrance tells you the probability that an individual will express a particular genotype. And with expressivity, that's more descriptive. So do you see a range or varying intensity of phenotypes? Or does everyone with a certain genotype pretty much express the phenotype in the same way, constant expressivity? If you learned something about penetrance and expressivity, please give me a thumbs up so that other students can find my content as well. And in the meantime, check out my website for tutoring resources, subscribe for even more biology content from me, and I will see you next time.